Thank you, Mr. Chairman, and thanks everyone for bearing with us. And I guess I'm the one keeping you from lunch, so I'll try to speed it up. And what I will be talking about is NicVax, which is a little bit different from what you've heard about so far today. It's first, it's a therapeutic vaccine, but it's one with a twist to drugs of addiction, which is a little bit novel in that arena. So, without further introduction, I'm just going to talk about the smoking in general, and this is not new to anybody here. It's clearly a great medical need to find ways to help people stop smoking. It's a very, very severe addiction. 1.3 billion smokers worldwide. In the US alone, 45 million adult smokers. And the cost of the healthcare system is just enormous, 192 billion direct and non-direct, of which about 90 billion or so direct cost to the healthcare. This is what the nicotine molecule on the far left, on your far left here, looks like. And two things that are extremely important for that small molecule. Number one, because of its size, because it's a very small molecule, about 162 daltons, it freely crosses the blood-brain barrier, which is important in how it actually develops the addiction, because then it binds to the receptors there. But importantly, because of its small size as well, you and I cannot produce antibodies to it, cannot protect ourselves. So even though it's foreign, we don't recognize it. Our immune system doesn't recognize it. So what Nabi has done is similar to what other conjugate vaccine technologies have done, which is taken a rather non-immunogenic molecule and then conjugate it to a carrier protein, a foreign carrier protein, that then elicits antibodies not only to the carrier protein, but also to the conjugated molecule, in this case, the hapten, which is the nicotine. So this is what we've done, and we use REPA, which stands for recombinant exoprotein A from Pseudomonas aeruginosa, which is very, very immunogenic. And we have at the end here, if you want, the REPA around which you find the nicotine molecules decorating it, so to speak. Some slides are missing, but we'll go through quickly the rest of it. I mean, certainly one of the things that the FDA and others have asked us for is, well, this nicotine, which binds to the same receptor as other very important molecules in the body that are extremely useful, have some similarities in structure, and therefore, is there any cross-reaction between the antibodies you raise to the nicotine and those important molecules? So we've ran a series of cross-reactivity experiments to show that the antibodies are rather specific to nicotine. Even the closest metabolite, which is cotinine, the cross-reactivity is infinitesimally small. So, okay, so let's talk about the mechanism of action and tell you what happens. As I mentioned, because of its small size, it freely crosses the blood-brain barrier and causes the nicotine the nicotine itself then attaches the nicotinic receptors, causes the release of dopamine, and that's what happens for the addiction. Because you release dopamine, you get this euphoric feeling from smoking, so you continue to smoke more. Our body actually reacts to that by upregulating the nicotinic receptors, and then at the end, therefore, you have even more of those receptors to bind more of the nicotine, and the cycle continues. What we have done, as I'm saying, through the conjugation is after the conjugation, now you get the antibodies to the nicotine. It binds the nicotine in the bloodstream and then prevents it from crossing the blood-brain barrier here, which therefore stops it, sequesters it in the blood, and prevents it from accessing the brain at all, which is a little bit different from everything we know about other smoking cessation therapies that are available in the market today, including nicotine patches and gums, but also Chantix and other things, and all of these molecules, therefore, act directly in the brain And therefore, safety becomes a question here. Safety is not at all a problem simply because you're sequestering it and leaving it inside the bloodstream as opposed to crossing the brain. We've done some early animal studies I'm going to walk through quickly, certainly as I mentioned to you, because of the mechanism of action we just talked about. Therefore, in essence then, you would imagine that when you have the antibodies in rats, in this case, if you test to see the nicotine after administration of nicotine in the bloodstream and in the brain, you would imagine that there is less going into the brain after immunization with NicVax versus placebo, and that's exactly what we see. If you look here in the open bars is the control, and in the hatched bars or the dark bars, the vaccine. You can see in the serum, in the control, very little remains after injection of nicotine, while in the brain, the majority of the nicotine is there. Conversely, in the vaccinated group of rats, in the serum, much of it is there, and in the brain has less of it in it. The question then becomes, if it's all in the serum and we are sure that that is due to the antibodies, you would imagine, therefore, if you look at the nicotine there, that the nicotine would be bound as opposed to free nicotine, because the antibody bounds it, and that's why it's sequestered in the bloodstream. And that's exactly what we see. Here in the control, whatever residual nicotine in the serum is actually most of it free, whereas the immunized group of rats, much of it is actually bound and only very little bit of it is free. So then we know now, at least in rats, that that is what happens. The other pharmacological and important elements that the antibody does to nicotine is actually it elongates the half-life of nicotine in the serum, in the blood. That is important because then it keeps the nicotine for a longer period of time and may have an impact on withdrawals and craving symptoms as well. These are some additional effect of the antibody of NicVax and the antibodies to nicotine on pharmacological effects of nicotine. It reduces the blood pressure, locomotor activity, nicotine discrimination, and also diminishes the nicotine self-administration. NSA stands for nicotine self-administration, which is a very, very popular model for drugs of addiction here. Okay, we'll move on then to what we think the hypothesis is, and this is just a hypothesis. Obviously, we don't have any proof of that's exactly what's happening, but it's just a working hypothesis based upon which we designed the clinical trial program. So if you bear with me here, somebody's smoking cigarettes here, and these, let's say, if you count them, it would be about 15 cigarettes or so. So it's not an important number of cigarettes. The point is, however, that the brain sees those cigarettes. As you continue to smoke, the brain sees the nicotine going in there. If you then imagine that we immunize somebody with NicVax, and those arrows here represents the NicVax immunization, you can see that as the antibody rises, the brain sees less and less of the nicotine, in essence. That is therefore important that we reduce gradually the dependence or the, if you want, the tolerance of the brain to the nicotine. In essence, we wean the brain slowly off the nicotine. And then at some point in the future, then we tell people to stop, target quit date here in the red arrow, and hopefully then we get successful quit. That is just a working hypothesis. Just want to make sure that you understand that that is the case. Now, with that in mind, we then went into the clinical development program. We've done a series of phase 1-2 clinical trials in humans, both smokers and non-smokers, and then moved on to a phase 2B proof of concept clinical trial, which we've conducted a few years back. This was a multi-center randomized double-blind placebo-controlled study in smokers who wanted to quit. 301 heavy smokers randomized 2-to-1 to NicVax and placebo, and we actually in that experiment, in that clinical trial, we tested 2 dose levels, 200 and 400 micrograms, as well as 4 and 5 injections in that proof of concept. First, to discuss the safety and tolerability, most common local reactogenicity events were aches and tenders, not unlike any other vaccine. most common systemic reactogenicity events with general discomfort, malaise, headaches, and muscle aches. Importantly, the events were generally mild to moderate in severity and resolved rather quickly. And the incidence of events did not increase after each immunization, which is obviously something that is important for us to recognize as well. There was no difference between placebo and any of the adverse events in each of the NicVax dose groups. And similar observations for all other studies to date were consistent. And importantly, when you compare the adverse events profile with that of other vaccines who are alloadjuvated, because NicVax is alloadjuvated in adults, you find that the adverse events profile is actually quite similar. This is just a very, very brief summary table. Here, upper respiratory infections, 29 versus 30, and you can go down the list. There is absolutely no difference between placebo and the NicVax group. Just one comment of the upper respiratory infection here. You can see that it's actually that we had this trial done in the winter time, and in the areas where done, this is not uncommon percentage of upper respiratory infections in there. Just to go to the antibody kinetics and see what it looks like. In essence, here's the 2 doses in 2 schedules. The blue is the schedule with 5 immunizations, as depicted by the blue arrows down there, and the 4 injection dose schedule was the 4 red arrows, and you can see that certainly the 5 injection schedule gives you a better profile of antibody and much higher responses. Again, the 400 versus 200, the 400 is a solid line, and the hatched line is the 200 microgram dose, and you can see again the 400 gives you a better immune response than the 200 micrograms. Based on that, we decided to move on with the 400 micrograms 5 immunization schedule. Before I move on to that, however, I just want you to notice, and I'll talk about it later on, we can see here that before you reach maximum antibody levels, there is a dip here, which is not uncommon in any vaccination schedule, but we wanted to find a way to smooth that as we move on, and I will tell you about it a bit later on, but just keep it in mind as we go on. One of the things that we wanted to make sure, and as we mentioned earlier, that the antibody response was very, very specific to the antigens, to the nicotine, and did not have any cross-reactivity to other antigens of similar structure. So what we wanted to do is to test the hypothesis that the antibody we raise may in fact be akin to a monoclonal antibody as opposed to a polyclonal antibody. And that's not very difficult to imagine, simply because the molecule is so small, as we talked about, that you can imagine that there is very, very few ways that the antibody can interact with that molecule. And in fact, this is the case. Here you can see that in immunized subjects that you can actually, in essence, have a monoclonal antibody that we have raised in mice. You can neutralize all of the antibody that you get from humans in there. So it's in essence, then you can see that the antibodies we raise are more or less monoclonal in nature. Then we get to some of the efficacy, proof of concept efficacy. It's not obviously efficacy, but some of the proof of concept observations that we had that is important for us to know. Here, this is actually, in essence, a point prevalence. That is not a regulatory term whatsoever, but it is an important, if you want, sensitive way to measure what the response would look like. So this point prevalence is only 1 week of continuous abstinence, just 1 week. Again, it's not a regulatory important determination whatsoever, but it's a sensitive one. And just before I explain the slide further, I just want to make sure that we understand that the proof of concept that we went into was that those who attain high antibody levels in response to NicVax have a better chance of stopping smoking. So the higher the antibody, the higher your potential of stopping smoking. And because of that, by protocol, we stratified, we indicated in the protocol that we stratify by high antibody versus low antibody group. And we chose 30% of the high antibody group. So in essence, when you look at the slide now, this blue solid line here represents the high antibody group versus the low antibody group and the placebo. And again, the low antibody group you can see here it didn't do any better than the placebo, and for a very important reason, and that is by protocol, anybody who drops out of those trials is considered a smoker. And because they drop out of the trial, their antibody is lower than the other group, and therefore they are all, in this case, regarded as smokers. Okay? Now, in these trials, which is a little bit different from other vaccine trials, the dropout rate is rather large. It is very, very high in those kind of trials. And in this case, we had about 35% or so. Just by comparison, other trials of all other drugs of abuse, including nicotine, are very high. And you know, the Chantix, for example, for Pfizer, was the dropout rate was something in the neighborhood of what we're talking about 40% or so. But that's typical of all of these kind of trials. Okay, this is the primary endpoint, and the primary endpoint was 8 weeks of continuous abstinence from week 19 to 26. That is the primary endpoint, in essence, in that yellow box here. So here's the high antibody group, 25% versus a placebo of 13%, with a statistically significant result here. Importantly, when you look at the long-term abstinence here was 18% for the high antibody group versus 6% for the placebo group here. That is 44 weeks of continuous abstinence, which is obviously important in this population simply because it's rather easy for somebody to stop smoking for a period of time. It is much more difficult to stop for a long period of time. And I mean, the ongoing joke in the industry is that what Mark Twain said, you know, stopping smoking is very, very easy. I've done it 100 times before. And it's really the problem why people stop so many times and start again. Okay, so let's look at the vaccine dose and schedule dependence of absence. So just about the ITT, and if I can, you know, in essence, focus your attention to the 5 injections because that's the regimen and that we're going with in a phase 3. It's actually 1 additional injection than in a phase 3. We actually have 6 injections, but it's closer to the phase 3 than what we have here. And if you look at this, now this is ITT, not stratified by high antibody and low antibody. And you can see that here in the NicVax group, in that group here, 16% versus 6% of the placebo. Again, statistically significant, so the odd ratio of 3.44. Here's an interesting analysis that we've done, which is actually quite important for us to take a few minutes to describe it. Again, this is high antibody versus low antibody and placebo. But in this case, we're actually looking at 8 weeks of continuous abstinence at a minimum. But then those folks would have to then continue abstaining till the end of the trial, to the 52 weeks. So it's not only 8 weeks, it's 8 weeks and then continue to the end. But the importance of that analysis is that it picks up people along the way who spontaneously, once they hit the antibody level that they need to help them stop smoking, can then stop. You can see that that happened in the high antibody group. So for example, here, a group of subjects who stopped in the middle, even though the target quit date was week 5 here, spontaneously on their own, they stopped. Here another group, here's a group. So in essence, once they reach the antibody, people's tolerance or people, if you want, threshold for ability to stop smoking has been lowered enough that they can stop on their own if they're willing to stop. This is a dose response, and dose here, it's not the dose of the vaccine, but it's really more the antibody levels that subjects have reached by 10 percentile. So this is the highest 10 percentile. This is the highest 20, the highest 30, and so on. And you can see there is a very, very decent relationship between the high antibody group. The higher the antibody, the better your chances of stopping. And that's what this is showing. In essence, it's here is showing the the percentile, and here's the, in essence, the 8 weeks of continuous abstinence rate. So it shows you then that the proof of concept that we went on to try to prove, which is the higher the antibody, the higher chances of success, has been shown in here. The other thing that we've done is looked at after every dose and look at what happens after every dose. Do we really get the same effect after every dose? And lo and behold, the same correlation holds true. After the second, third, or fourth injection, you still get, the higher the antibody, the better your chances of stopping smoking. One additional observation that we were actually asked about by the regulators is what happens to those who may oversmoke. It's not a difficult concept for one to imagine, because I'm giving you a vaccine that raises your antibody and gives you higher antibody, in essence, that then prevents you from enjoying a cigarette. What one may do is to oversmoke, smoke more, so they can overcome the antibody. So that's the question that the regulators asked us. What we found is actually exactly the opposite, and we can imagine this theory after we found the observation. But here, the higher antibody group, remember that 30% we talked about, and these are the number of cigarettes here versus weeks in the study. And you can see that they actually had a lower number of cigarettes consumed versus placebo and the low antibody group. So it's exactly the opposite of one would have imagined. Actually statistically significant, highly statistically significant. So even for those who continue to smoke on NicVax, they consume less cigarettes. That may be clinically important, although the regulators obviously would like better evidence than this, but it then reduces the harm if you are able to reduce the number of cigarettes that you're smoking. Remember I told you about that dip in the antibody profile that we didn't like in the beginning with the 5-dose immunization schedule. We then tried to find a way to overcome that dip by adding another injection. Remember, we started off in the phase 2 proof of concept with 5 injections. We said that's the schedule that we like the best. Well, what we've done here is we added an additional injection in another phase 2 trial that was not a smoking cessation trial, was just simply an immunogenicity trial, and then asked the question, what happens if I add an additional dose at week 12 at the time we see the dip, which is now what we would like to have as the target quit date on week 14. So we're asking people to quit at week 14, and therefore we decided to add another injection at week 12. So instead of 0, 4, 8, 16, and 26, we have 0, 4, 8, 12, 16, and 26. And if you look at this, then here you can see then we have been able to smooth that dip here. In the blue is the proof of concept 5 injection schedule. In the purple is a 6 injection schedule, as I'm saying in that other phase 2 trial that we have conducted. So in essence, we smooth that. And now if you look, we also move the target quit date from week 5, as I mentioned earlier, to week 14. At week 5, and the reason for that is very simply, at week 5, we had very few people, here is 7% who attained what we considered to be the antibody level that may help people smoke. In essence, if you then move it to week 14 here, you can see that the majority of people have the antibody level that we think may be helpful for people to quit. I'm just going to tell you then that the last few minutes of this talk is just very simply where we are with the phase 3. We have 2 phase 3s ongoing, and just here's the comparison between the phase 2 and the phase 3 design. In essence, the 5 injections in the proof of concept study versus 6 injections in the efficacy study. Target quit date, as I mentioned, moved from week 5 to week 14 to better align the high antibody level with the target quit date. Obviously the trial sizes, we have 2 trials of 1,000 participants, a randomized 1-to-1 placebo NicVax, and clearly many more research sites. And in smoking cigarettes, of 15 cigarettes, we had in the proof of concept, which was not exactly what the FDA is used to, and we discussed with the FDA and they actually agreed that the 10 cigarettes is what everybody else has been doing so far, so that's what we're doing. One important thing that, that certainly Pfizer has pioneered with the Chantix study is related to the counseling, behavior counseling, which is extremely important for that, and we also have that standard of care behavioral counseling as well. These are competing vaccines, and where they are, I'm not going to dwell on this. Needless to say that all other vaccines in the arena for smoking cessation are still in phase 2, and they have not progressed to phase 3 yet. So NicVax is because of that, obviously in a leading position. So we had an agreement with GlaxoSmithKline here, and the reason we've done that is that obviously GSK has probably not only the vaccine experience, but they also have the smoking cessation experience, both over-the-counter nicotine patches and gums, but in addition to that, they also have prescriptions IBAN, so because of that, they were the ideal partner for us. We are risk sharing with them, so in essence, we are developing the first generation NicVax to which they have an option, while they are developing the second generation using their very potent adjuvants that they have, which therefore is a very important thing for Nabi to consider. Clearly, it's an exclusive worldwide option license agreement, and these are the agreement terms. Clear, it's actually public information all over the place. Unless somebody is very, very interested in a question that I can respond to anything like this, I'm happy to take questions based on the agreement itself. And with that, just a very brief where we are in the pivotal study. As I'm saying, 2 1,000 subject phase 3 studies. First one was the first patient was randomized November 2009, the second March 2010. Enrollment is actually, should say complete here for both trials. Both trials are now fully enrolled. We anticipate results from the first phase 3 trial in the second half of this year, and results from the second trial in the first half of next year. And with that, I'll close in the sake of time, and I'll take any questions if there is any. Thank you.
Thank you, Dr. Fahim. I'll release everybody for lunch, but those who have a couple questions, we'll go take 2 and then I think adjourn.
You have talked about the high antibody production, but not about the age of the individuals who participate. So how good is a chance of an elderly patient to develop high antibodies after the vaccination?
If I understand the question correctly, you're saying the age,
yes,
the age of the participants, yes, and their antibody levels. So actually the age group in both trials, the phase 2 and the phase 3, is 0 to 65. 18 to 65, not 0. 18 to 65 years of age. And there was, we stratified by age, and there was no difference in antibodies between them. So everybody responded very well, including the upper limit of the age, responded very well to the vaccine. So there was no difference in age.
Thank you for the presentation. 2 questions. So first question, now that you're working with GSK, they have a nicotine patch for those subjects that do develop antibodies and then have cessation of smoking. But for those subjects who may want to put a nicotine patch on, so what's the safety answer on that? And the other is, as the titers seem to wind down off the last injections, what's your vision in terms of continuous reimmunizations, and how long apart would you imagine that to be?
So the 2 questions are actually extremely interesting. To answer the first one, what happens when you get a patch when you have antibodies? Obviously, there is no side effects whatsoever, because there is no reason to imagine side effects. You're giving nicotine, whether it's from a cigarette or a patch, doesn't matter to the body. But if you take the patch at a very, very high antibody duration, you may neutralize much of that patch, because the nicotine from the patch will be neutralized just as much as the nicotine from a cigarette. So that's the answer to the question. So unless you give it at the time where the antibody is still low, it probably wouldn't have the same effect. You can over give a patch, but certainly Is that a follow-up question?
Well, the question was around whether or not you didn't expect a reaction around the patch.
So I answered that initially by saying there is no reason to imagine note, and we have certainly in the clinical trials we've done, we didn't allow people to take the patch because that defies the whole purpose of it. So I can't answer it directly, but there is no scientific reason to imagine so. So, and then the decline in antibody very quickly, clearly like any other vaccine, there is a decline in antibody. But the important thing to bear in mind is the following, that as you are smoking, you need a very, very high antibody level to neutralize the nicotine that you're getting. When you stop smoking, for maintenance, long-term maintenance, you don't need as much antibody, which is not different from any other vaccine. Again, if you are attacked by a virus or bacteria, you need a high antibody to neutralize it, but if you're in maintenance mode, you don't need as much. Having said that, your question is, can we boost them? And there is no reason to imagine that you cannot boost them in the future.
Well, let's thank Dr. Fahim and all of our speakers this
Thank
morning
you.
again.